Hi there, welcome back to the channel. I'm the GCSE science teacher, and in today's lesson, we'll be learning about microscopes for the GCSE Biology course. Content covered today includes microscopes, required practical method, and microscope calculations. Let's get started. So, what actually is a microscope? It's a device which is used to view objects which are very small and we cannot see with our own eyes. You've probably used a microscope before, and this would most likely be a light microscope. How does this work? Well, it manipulates light which enters the eyes using a convex lens. And those convex lenses bend light to actually magnify that image. Just as a run through what a light microscope looks like, we can see an image here. Eyepiece lenses will have a times 10 magnification, and we use the eyepiece lens to view the object. The objective lenses are used to further magnify the image, and they can have a times 4, times 40, or times 100 magnification. So, looking at this example, we could have a total magnification of times 1,000 if we wanted to by using the times 100 objective lens and then the times 10 eyepiece lens. The stage itself is used to place the object we are looking at on so that it doesn't move around too much. And the coarse and fine focus knobs are used to actually clarify the image and actually produce a clear, crisp image as well. Below the stage, you'll find a light source which can be used to shine light through the object we're looking at. And that's why anytime you use a microscope, the actual object you're looking at has to be very, very thin so that light can actually pass through it. So, microscopes have been around for a really long time, but we're unsure as scientists who the originator was in terms of this brilliant device. However, we do know in the early 1600s there were some scientists who did come up with similar devices, and that's where we think they originated from. So, the Dutch specular maker, the spectacle maker, I should say, Zacharias Janssen, he was one of the first scientists who came up with the idea of microscopy, but also Luke Hook, who was now known as the father of microbiology because of his amazing work with discovering protists and bacteria and other cellular organisms. At GCSE, you need to be aware of light microscopes and electron microscopes in terms of the positives and negatives of each device. So let's talk about those now. Light microscopes are portable, less expensive. They can show true color, but we can also use stains on those specimens in case the object does not have a color. Those stains are also used to actually view specific organelles as well. With a light microscope, you can use organisms which are living. So that means we don't have to destroy any tissue, and that can be very beneficial, especially if we want to look at insects. However, with a light microscope, we cannot see ribosomes, and that's why electron microscopes have been utilized, especially in research, to actually see subcellular structures like ribosomes, which are very, very small. Electron microscopes have a very high magnification and resolution. Resolution, by the way, is the ability to distinguish between two specific points clearly. So imagine you've taken a picture with your phone and the image is all blurry. We would say that has a low resolution. If the image, however, was very clear and we could zoom in and see all the different pixels in very clear clarity, we would say it has a high resolution. However, electron microscopes are very expensive and are often only found in research institutions like universities. They do also produce only black and white images. Although they are 3D, those images don't show any color, which is why often we have to then go in with specific software to add color in. The specimens are also negative of using electron microscopes. So, in terms of the required practical, one of the key things to note whenever you're writing a method for a required practical is that you want to imagine that that method is going to be given to someone who has never done the practical before. That way, it will mean that you've written in a very clear, concise way so that the person reading it could make reproducible results. That's how you're going to get into the higher level marks for your answer. So when you're doing this practical, you want to use a pair of tweezers to peel a very thin layer of the specimen. Often it's described as the onion practical. It doesn't have to be the onion. It could be any type of cell. It doesn't even have to be plant tissue. You then place this specimen onto a glass slide, and it has to be a clean slide without any fingerprints on so the image is nice and clear. You then want to add some stain so you can see the organelles very clearly. The stain could be iodine, for example, but there are others that you could use. You then want to place a cover slip over the top using a mounted needle, and the mounted needle is used to lower the cover slip slowly so there are no bubbles that could actually obstruct the view of your specimen. You then want to place the slide onto the stage and use the lowest objective lens first. Adjust the coarse focus knob to bring the stage closer to the objective lens. And this way, when you view your specimen by looking down the eyepiece lens, you're not going to cause the stage to actually move any closer. You're actually going to push the stage downwards by turning the focus knob. And that way, you're not going to have the problem of the slide and the objective lens clashing together and colliding, which could be a big problem. Once you have a clear image, you want to draw the image using a pencil and you want to label the image clearly. Remember, if it's a light microscope, you cannot see ribosomes, so be careful not to include that in your answer. You then can also calculate the magnification, object size, and image size of the specimen. So let's do that now. So, when you're doing calculations with microscopy, you need to remember I am. This is one of the best ways that I've found teaching this. A lot of students forget which bit of the equation they need to know. But just so you remember, the image size is given the symbol I, the actual object size is A, and the magnification is M. Remember, the actual size is going to be very, very small because it's how big the actual object is. The other thing I've noticed when marking work is students often forget to calculate or convert the units. So if you are going from millimeters to micrometers, you want to times the answer by 1,000. And if you're going from micrometers to a millimeter, you want to divide by 1,000. The key to this is to convert your answer first before you put it into the equation. You also can use the triangle method to rearrange the equation, but just remember not to rely always on the triangle method for rearranging, especially when you get more complicated equations. So let's do an example. Calculate the magnification of an object which has an actual length of 5,300 micrometers and an image size of zero point 0.5 millimeters. First of all, you want to write out the equation you'll need. So we're trying to calculate magnification. So m equals I divided by a. You then want to convert your units. So we're going to get everything into micrometers. So if we do zero point 0.5 millimeters times 1,000, we should get an answer of 500 micrometers. You then want to put your answer or your numbers into the equation. It's always important to write every step you do that you put into your calculator onto a piece of paper so there's evidence of your working. Once you do 5,300 divided by 500 in this case, you get an answer of 10.6 times. And because it's a magnification, we just use the times symbol to show that it's a magnification. I hope that was helpful. I've been the GCSE science teacher and you have been curious. If you did enjoy that video, please feel free to like, comment, share, and subscribe. And if you want more GCSE content, feel free to follow me on Instagram at the GCSE science teacher. You can also comment below if you have any topics you would like specifically covering here on my channel or on Instagram. And yes, I hope that was helpful. If you do have any questions, feel free to comment. Until next time, have a great day. Bye.